Hello. We've seen that electron transport and oxidative phosphorylation of ATP are coupled processes that can be chemically and even physically separated. In this module, we ask the question, how are the electron transport system and oxidative phosphorylation physically coupled? This schematic suggests a simple mechanism predicting an enzyme that would catalyze a substrate-level phosphorylation of ADP coupled to a very spontaneous redox reaction in the electron transport system, much like the coupled reactions we saw in glycolysis. Despite looking for such enzymes, none were found. What was found was a role for all those protons that are generated by reactions forming NADH and H+. In those reactions, NAD+ is an oxidizing agent and pulls a hydrogen molecule off of the molecule being oxidized, but then splits it into a hydride ion, which binds to the NAD+, and a free proton. This slide shows what happens when electrons flow from NADH, or FADH2, to oxygen down the electron transport system. As electron carriers are oxidized and the electrons move down the electron transport chain, the free energy released powered proton pumps in the crystal membrane that pump protons into the space between the crystal membrane and the outer membrane. Some protons are shown in the cytoplasm in the drawing because the outer membrane, or OM, well, it leaks protons. But the bottom line here is that proton pumps have used the free energy released by the electron transport system redox reactions to create a gradient of protons. So, a proton gradient, in fact any gradient, stores free energy. It follows, then, that relief of the gradient will release free energy. Let's look at a prediction and a test of the hypothesis here that relief of a proton gradient is coupled to ATP synthesis in mitochondria. Here's one prediction. When isolated mitochondria oxidize NADH, or FADH2, the pH of the solution around the mitochondria should change relative to inside the mitochondrion. Here's the test. If respiratory substrates are added to the mitochondria, specifically NADH and oxygen here, the pH of the solution outside the mitochondria should drop as the solution accumulates protons pumped out of the matrix. That solution becomes acidic. Note that for this experiment to work, the solution had to be unbuffered. When this experiment was actually done, the pH of the solution did drop, supporting the chemiosmotic hypothesis. Here are some more ideas for testing the hypothesis, so think about these experiments. Let's repeat the experiment using Racker's studded vesicles. What would happen? Then repeat the experiment using his smooth crystal membrane vesicles. What would happen then? And here's a challenging one. Since electron transport can be separated from ATP synthesis, can you come up with an experiment to show that mitochondria can actually make ATP in the absence of electron transport? This slide lists some properties of the crystal membrane. You'll see over the course of this class that the crystal membrane has somewhat unusual properties compared, say, to a plasma membrane. So it contains more than 70 different polypeptides, membrane proteins. The proteins comprise about 75%, actually more than 75%, of the membrane mass. They contain a high concentration of the phospholipid called cardiolipin. It's a very saturated phospholipid, which means that crystal membranes are somewhat less fluid than other membranes. You'll learn the effects of saturated fatty acids on phospholipids on membrane fluidity when we cover membranes in a later module. The electron carriers themselves comprise 15 proteins, as well as a non-protein organic molecule, coenzyme Q, that we saw before. The electron transport proteins are not organized in any particular order on the membrane, so that the rate of electron transport really depends on the concentration of reduced electron carriers, the number of these electron transport proteins, and the rate at which these proteins diffuse laterally in the crystal membrane. When isolated from the crystal membrane, most crystal membrane proteins lose any biological activity that they might have had. But it is possible to isolate phospholipid protein complexes that do retain biological redox activity. This can be demonstrated by adding electron donors or reduced substrates to these complexes In the presence of what's called a redox dye, now this is just a chemical that turns color when it accepts electrons. So the idea is that the substrate will donate electrons to some phospholipid protein complex, which will in turn donate those proteins to a dye that will change color, allowing you to see the reaction. These kinds of observations led to the conclusion that the proper folding and shape of electron transport proteins depends on their correct association with crystal membrane phospholipids. Here's a cartoon of the crystal membrane showing the respiratory protein complexes as well as several other membrane proteins whose function is known. At the top are the respiratory proteins or electron transport protein phospholipid complexes, labeled complexes 1, 2, 3, and 4, along with coenzyme Q. Again, these are shown organized so that you can watch the electrons flow, but in reality, they are not organized and held together in any form on the membrane. They have to diffuse and bump into one another in order to take electrons one from another complex. Complexes 1 and 4 and coenzyme Q use the free energy of electron transport from NADH or FADH2 to force protons out of the mitochondrial matrix. Because this process requires energy, you may recognize it as a form of active transport. The other crystal membrane proteins shown here are all involved in one or another form of transport of materials across the crystal membrane. Uniport brings pyruvate into the mitochondrial matrix. Antiport brings ADP into the matrix while exporting the newly synthesized ATP to the cytoplasm. Cotransport uses some of the accumulated positively charged protons to get negatively charged phosphate ions into the matrix without changing the net charge in the matrix. And finally, in another example of uniport, protons flow back into the matrix through the F1 ATP synthase particle, here it looks vaguely like the lollipop that we talked about earlier, to relieve the proton gradient. We're going to take a closer look at this last process in just a second. So, we can demonstrate proton pumps, but what are the proton pumps? I alluded to the fact that they're part of the phospholipid protein complexes. This slide illustrates the preparation of synthetic phospholipid bilayers, that is synthetic membranes, in which one of the respiratory complexes was inserted, in this case complex 1. When NADH is added to these membranes as an electron donor, along with the compound X, one of these redox dyes, to accept electrons from the complex, protons were seen to be pumped into the vesicles so that the pH of the solution increased. This shows that complex 1 was not only a redox complex, but a proton pump. These experiments also showed that there were 3 sites of proton pumping, and that these sites, complexes 1 and 4 as well as coenzyme Q, correspond to the 3 steps in electron transport that we saw earlier, involving large negative standard free energy changes that were originally thought to be actual sites of ATP synthesis. We even know something about the stoichiometry of proton pumping. About 4 protons are pumped across the crystal membrane for every pair of electrons transferred from complex 4, also called cytochrome oxidase, to oxygen. Now, here on the left is an electron micrograph of a single F1 ATP synthase molecule. As promised a moment ago, let's look at our current understanding of how ATP synthase works. First of all, it's a regulated proton gate, controlling the flow of protons into the mitochondrial matrix from the intermembrane space, or from outside the mitochondrion. Note the multiple polypeptide subunits. Some are embedded in the crystal membrane. These are called the F0 part of the structure. Others make up the lollipop head, the F1 particle facing the mitochondrial matrix, that Racker isolated. This head consists of alpha and beta subunits. When there's a lot of ADP around, which would indicate that the cell is rapidly using up ATP, so this is a busy cell, the excess ADP binds to the alpha subunit and opens up the proton gate. So the alpha subunit is a regulatory protein whose shape changes when it binds this excess ADP, opening a pathway for the protons to flow back into the matrix from where they have accumulated during electron transport. When the gate is open, the protons in the intermembrane space of the mitochondrion bind to the A subunit shown in purple here. The C subunit forms a cylinder that can spin like the armature of a motor when protons are passed to them from the A subunit. As the armature spins, protons are ejected into the matrix and ATP is synthesized. as you saw repeated here a few times. The process of active transport that pumps protons, followed by the regulated relief of the proton gradient through a proton gate, is the essence of oxidative phosphorylation. As summarized on this slide, the pumps are coenzyme Q and respiratory complexes 1 and 4, as I mentioned. They are fueled by the negative free energy change of their redox reactions, and the F1 ATP synthase is a regulated proton gate using free energy stored in the proton gradient to make ATP. Think about why these gates are regulated. So finally, let's look at a last balance sheet for respiration. In the end, 36 to 38 ATPs have been synthesized. 4 by substrate level phosphorylation, and between 32 and 34 by oxidative phosphorylation. The difference, by the way, of 2 ATPs from oxidative phosphorylation is because there are actually 2 shuttles for getting electrons on cytoplasmic NADH into the mitochondrial matrix, one of which donates the electrons to carriers further along the electron transport chain and therefore permit fewer ATPs to be synthesized. 6 CO2 have been made, 2 from pyruvate oxidation outside the Krebs cycle, and 4 CO2s that are made in the Krebs cycle itself, again per 2 turns of the cycle. Remember that the phosphorylation of ATP is a condensation reaction, so that 36 to 38 waters have been produced by the sum of substrate phosphorylation and oxidative phosphorylation. And finally, here is the chemical equation for the whole of respiration, including ATP synthesis. So can you find the 6 water molecules that result from glucose combustion? This module on oxidative phosphorylation then concludes our discussion of respiration.